Awesome. Well, welcome. My name is Kayla Clark, and I coordinate the public programs for UGA Marine Extension and Georgia Sea Grant. And as an organization, we do outreach, extension, research, and education with the goal of contributing to healthy coastal ecosystems and communities. And one of the ways that we do that here at our education center is through citizen science and through engaging the public and monitoring our local waters. So I'm really excited that you'll get a chance today to talk with Katie Higgins, who is not only a marine educator, but also our volunteer coordinator. And she manages our phytoplankton monitoring network, which is a citizen science project that you'll learn more about today. In addition to the citizen science, we also do a lot of fun education, summer camps, public programs. And so Madeline McDonald is a Sea Grant Marine Education Fellow. And normally this time of year, we would be in full swing with our on-site summer camps. So the Great Plankton Sync Off is actually a modified activity from those summer camps. And I would encourage anyone on the line to check them out in future years coming up as well, and think about joining us for those. You will also meet Ipsita Tingy, who is my colleague in helping coordinate these public programs this summer, and she'll be helping with moderating as well. So with that, I'm going to go ahead and throw the ball over to Katie to get us started with what a plankton is and why they're important.
All right, well, thank you, Kayla, for that introduction. And it's really nice to have you all with us virtually here. So we're going to talk a little bit about plankton, and I'm going to do my best to give you a general overview of what plankton is, talk about differences between phyto and zooplankton, and also introduce a little bit about our citizen science phytoplankton monitoring network group. So we're going to start by just throwing it out to you and getting an idea of what your thoughts are, what you already know about plankton. So go ahead and use your chat box and let us know anything at all that you might know about plankton. I'm going to give you just a minute here to see what you have to say.
So we do have an answer from John saying that they are often the bottom of the food chain.
Okay, excellent. Yes, they are definitely the base of the ocean food chain. So we're going to talk a little bit about that, but as such, they play a big role in cycling nutrients throughout the ocean. Anything else from our chat box?
That many plankton are microscopic, from Catherine.
Okay. Okay, absolutely. You are correct. Quite a few of our plankton are microscopic. However, there are plankton that are much larger and you can see with the naked eye. As a matter of fact, several of them are very large. So don't be surprised if I throw jellyfish into the whole planktonic piece because as most of our plankton, you'll notice that I have here that the word plankton, the base of that word plank, basically means wanderer. So anything that cannot swim against the current is going to be considered plankton. So jellyfish also fit into that. But you're right, the vast majority of what we consider plankton are microscopic. Anything else, Kayla?
That seems like it. Nina had added that plankton can't swim, but you already touched on that.
Okay, excellent. Well, so we're going to talk about that general piece. Now, again, because we're talking about a lifestyle, so not being able to swim, that planktonic sort of lifestyle rather than a specific group of species. We can break our plankton into sort of two very large groups. One of them are the zooplankton and the other are phytoplankton. So our zooplankton, the word zoo sounds like zoo, and those organisms are mostly animal-like. You may have zooplankton that eats plant-like organisms or that eat other zooplankton or animal-like organisms. They're generally multicellular, so they're a bit more complicated than our phytoplankton on the other side. And they can break into two sort of major groups. So our zooplankton can be broken even further into what we call meroplankton, which you can think of as all of the organisms that may spend only a portion of their life as plankton. So they will metamorph or grow into a different type of organism. For example, this little crab zoea here will eventually change into what we know as that walking crab. So it will flatten out and it will metamorph, much like a butterfly changes from a caterpillar to a butterfly. So our meroplankton only spend a portion of their lifetime as plankton, whereas haloplankton spend their whole life. So again, that H sound will help you remember haloplankton as those organisms that spend their whole life as plankton. You can see that we have different organisms that are able to spend a certain amount of their time basically out there. Now, some of our plankton, often people will talk about the fact that there is a character in SpongeBob that looks like a plankton, and we certainly have some of our haloplankton that look like SpongeBob's plankton. That is a copepod there at the bottom, and it does spend its whole life as plankton. Our phytoplankton on the other side are plant-like plankton. So these organisms are they are primary producers. They use photosynthesis just like plants do. So they actually have little tiny cells in there that are called chloroplasts that produce chlorophyll and allow these organisms to produce their own energy just like plants. They're generally unicellular or they are single cell organisms. And we have, again, two kind of large groups. We have diatoms and we have dinoflagellates. Our diatoms, one of the easiest ways to sort of separate them is what their cell walls are made out of. Our diatoms are made out of silica, so they live in what we consider glass houses, right? Silica is the same chemical element as we have in glass. So diatoms live in glass houses, and dinoflagellates live in wooden houses. So their cell walls are actually made of cellulose. There are also some other pretty major differences in them, but we'll talk about it a little bit more as we get into our identification piece for them. But we'll be talking about diatoms and dinoflagellates. Now, during this program, I'd like to talk a little bit more about where they live in the water column. So just because they are considered wanderers does not mean that they don't have a preferred habitat. So if you think about it, if we look at our ocean habitat, even if you look at just the water column, we've got different areas in that water column that have physical properties that are going to be better for organisms. So if you think about our phytoplankton that produce their own energy using sunlight, they're going to want to live in that upper layer of the ocean that is considered our photic zone, or you can also call it the sunlit zone. That's where our sunlight will penetrate, and it allows these organisms to actually access that sun. So they do have some adaptations that allow them to float in the water rather than sink in the water. And we're going to get a little bit further into that as we continue our program. So anything that creates its own energy is going to need to be in that photic zone, but also any of our zooplankton that might eat that phytoplankton are also going to need to live in that upper layer of the ocean, as well as any other organisms that might eat plankton, that eat phytoplankton, etc. So thinking about how these organisms contribute to that food chain, as we mentioned, they are the bottom of the food chain. Even as these organisms die, they will sink down into the deeper portions of the ocean and feed animals and organisms that live in that benthic or bottom zone. So they will continue to cycle nutrients throughout the entire ocean. Our phytoplankton, which we're going to focus on a little bit more for this program, are an amazing group of organisms, right? If you think about it, not only are they that base of the food chain, but they also produce over 50% of all of the oxygen on the planet. So I want you to take a minute and I want you to breathe deeply in and out 2 times. Half of that oxygen that you just brought in is thanks to the phytoplankton on our planet. So we are very lucky to have them. Without phytoplankton, there wouldn't be life on Earth the way we think of it or the way we experience it today. They're also responsible for transferring a lot of the carbon dioxide from the atmosphere they take care of that carbon dioxide. So they actually consume carbon dioxide and they expel the oxygen, just like our trees and other plants. They can even affect our cloud formation and our atmosphere. They are also the base of many of the products that you and I use on a daily basis. If you cook with sea salt, you're going to end up with phytoplankton in your diet. If you use toothpaste, they often will use diatoms as the base, as sort of the scrubbing piece in the toothpaste to actually clean your teeth, that has that silica-based diatoms in there that help to clean your teeth. So many of the products that we use also contain phytoplankton. Now, just like most things, too much of a good thing can actually be an issue. There are such things as harmful algal blooms. You may have heard of red tides. And red tides are actually caused by a type of phytoplankton or algae in the water that gets a high number of nutrients. All of the things sort of coincide, so all those physical aspects coincide, and they can create blooms. It's a plant-based organism, so it can bloom, and that means that where we always have a certain number of these organisms in the water, they all of a sudden have so many organisms in the water that they can actually block the sunlight, so they can cause physical issues with water quality out there. Some of the diatoms and dinoflagellates, when they bloom, can actually be toxin producing, so they can create poisons in the water. They can also aerosolize or get some of those toxins into the air and cause problems for breathing. So one of the best known red tide situations most recently happened in Florida with an organism called Corynea brevia, which produces a brevetoxin. And when that brevetoxin is breathed in by humans, it can be very irritating. It can also cause problems with the immune system, with some of our marine organisms like fish and sea turtles and some of our other longer living animals. So harmful algal blooms can cause all kinds of different problems, including shellfish poisoning, etc. And that's where we get to our volunteer or our citizen scientist aspect of this. It's important to note that not all phytoplankton bloom and produce problems, right? There are a lot of times when phytoplankton bloom and they are a great food source. So they are not necessarily always going to be a problem just because there is a bloom. However, for those handful that can cause problems, our volunteers monitor the waters every week or 2 weeks, depending on the group. And they are trained to recognize a certain target group of organisms. So there's about 12 organisms that we work to identify here in Georgia. Some of those can cause physical damage when they bloom. This organism here on the left, Kashiwo, will actually produce a sort of soap-like soap-like product that can cover the lungs, or it can clog the gills of things like oysters or fish and can cause problems or die-offs. This Catastrophes here has sort of spiky little points on it which help it to float in the water, but when it blooms, those little pointy spiky things can actually cause damage to gills. Or we have other organisms here which will actually potentially produce oxalic acid, which can cause a lot of problems, irritable bowel, pretty extensive bowel problems. It can also potentially cause neurological or immunological problems down the road. So those are the types of organisms that we, working with the Phytoplankton Monitoring Network, will learn to identify. Now to do this, we actually use very simple equipment, but it's important to sort of see some of that equipment. So I'm going to share my screen a little bit bigger and show you some of the equipment that we use for the phytoplankton monitoring group. We use the refractometer here to determine salinity. So higher salinities can actually cause some of those blooms. So knowing our salinity in the water is very important. We also take the temperature and tidal point while we're out there looking at physical parameters. And then we use a special net. So you can see this is a small version of a plankton net, and we're able to tow this through the water just with a little rope here. We tow that through the water for about 3 minutes. This is a very fine mesh, so the water will actually go through the net, leaving microscopic phytoplankton behind. And then that water going through will actually concentrate it into this little bottle. So we have a very simple way to take that sample back to our lab. We then use an eyedropper and a gridded slide here so that we can be sure that we're methodical about going through that slide and identifying all of the organisms that we find. And then, of course, we use a microscope, a compound microscope, to be able to use to actually see those organisms. I'm going to go back to my big screen here because I have a short video that will allow us to go through one of the slides and see some of the organisms that we're going to work on identifying here. So I'm going to go ahead and hit play. I may hit pause as I go through here, so I have a few more minutes to talk about it, but you can see an organism here that is moving through. This is a small diatom. It's called a pleurosigma. And as you can see, some of our phytoplankton, even though they're plant-like, they actually move. So we have the pleurosigma here, which is sort of an S shape. You can see, oops, I apologize, meant to hit play again. So it's kind of an S shape on this particular cell. You can recognize it by that curvature at the end of each of the points there. We also have another organism here that comes in chains. So each one of these little dots is an individual organism, but they will form these chains. This one is called skeletonema, and it's named because it looks like vertebra. So it looks like your backbone, and it will form those very long chains. And you can see as we move through the slide, we're also going to get to the next portion of the slide here. But we'll go through, and we don't exactly count things, but we get a good idea of how many are in our frame. So if there's just a few, or if there are 20 or 30, or if there were a bloom, we would actually have so many that it would be hard to tell individuals. We're going to look at another organism here. This one is one of our targets, and it is considered one of those organisms that can actually produce okadaic acid. Oftentimes you'll see that it is a dinoflagellate, and the reason I can tell that is that it has sort of different parts to the organism. It has this little flange here at the top, and it has this constriction between the two portions of its cell. And in that constriction, while you can't see it, there is a flagellum that would be here, and it would allow this organism to actually move around quite a bit. It would also allow, you can see this other little flange here that kind of helps to keep it stable as it moves through. So this is one of our target organisms. All right, so I do think that this is as much as we can do as far as looking at the different organisms, and I want to open it up so that we can have a few question and answers as we go through. So Kayla, do we have any questions that are coming up in our chat box?
We had a question about whether or not the plankton, as it sinks to the bottom of the ocean, is it contributing to the ooze that's on the bottom?
Ooze, as in sort of the muddy bottom of our Georgia coast. It certainly does contribute to some of that, and maybe to some of that texture. The sediment itself is actually coming from our mountains and washing down our rivers into the Georgia coast. And because of our barrier islands, and because of the sort of large amount of ebb and flow on our coast, as well as a very shallow continental shelf, we end up retaining a lot of that sediment. So we end up having a very muddy shore here in Georgia. But certainly the organic material is contributed to by the phytoplankton in the water.
Thanks, Katie. We also had a question about the increase in harmful algal blooms and whether or not that's related to an increase in chemical pollution in the oceans.
I'm sorry, could you repeat the question, Kayla?
Is the increase in harmful algal blooms related to an increase in chemical pollution?
So it's certainly harmful algal blooms. All algal blooms do have a contribution with nitrogen runoff. If we're using a lot of fertilizer, or in the summertime, natural sort of increases to temperature in the summer, combined with a greater amount of human activity on our beaches and a greater amount of runoff in that season, certainly do contribute to those harmful algal blooms.
Thank you for explaining that. And we also had a question about the flagellum that you mentioned on that last organism. Can you describe kind of again what a flagellum is?
A flagellum is sort of like a projection. It's a hair-like projection, and they can move that through the water. So it will actually give them a little bit of movement in the water. And our dinoflagellates are named for that flagellum, and they all have that sort of constriction. Now they all look a little different, and all the organisms are differently, they have a different conformation of their body shape, but they all do have the flagellum. They'll actually have a couple of them, and it will allow them to move through the water.
That is so cool. And I have to say, it's really cool when they're underneath the microscope to see some of them moving around.
It is. Some of them look like little tiny dancers,
Exactly. We had another question about whether global warming affects algal blooms, specifically harmful algal blooms.
There are certainly concerns about as our oceans are warming, there is a contribution to harmful algal blooms. It's also potentially affecting a lot of those natural cycles for our planktonic cycles. So in some of our colder water areas where that upwelling and some of the circulation of our ocean water is changing. It's changing entire life cycles for some of the plankton out there. but certainly warmer temperatures can cause an extended period of time when blooms are happening as well. So, so there are certainly concerns about the increase, not only of, of some of our other issues as far as flooding and contribution to atmosphere. And as I mentioned, phytoplankton can actually affect our atmosphere and cloud formation and the water cycle itself. So, so there are some concerns that global warming we're going to see more often, we'll see more frequent hazardous algal blooms.
Well, and I think that that's one of the reasons I find the work that you and your volunteers do so important, is having that early detection system. So even if we haven't had large harmful algal blooms as often here in Georgia, it's nice to know that system is in place for the future moving forward too.
Well, and a good example of that is that the organism I talked about that creates the brevetoxin, so the Carinea brevia, which has bloomed in Florida and has traditionally been, that sort of Gulf of Mexico issue as far as those harmful algal blooms. Those have moved up further onto the Atlantic coast and further north. And so the last couple of years, we have been on the lookout to actually see that organism here in Georgia. So while it's on our list, we don't see it. The issue is that as waters warm, will that organism continue spreading north?
Well, these are great questions from the audience. Those are all the ones that I've seen so far. We have just a few more minutes left. Does anyone, before we transition to the Great Plankton Sync Off activity, are there any last questions that people are wondering about? And if there are, you can add them to the chat box. And it may be that we have some more filter in. So we'll be sure to leave some time for Q&A at the end as well. But I think at this point, what we're going to do is transition to hearing from Madeline about how we can take some of what we've learned in terms of plankton adaptations and run our own test using craft materials that you might have at home. And we'll add this in the chat box as well. But we do have activity directions. If you want to do the same activity that Madeline is going to lead us through, if you want to do that at home, you can find those activities in a downloadable sheet as well. So with that, I'm going to go ahead and turn it over to Madeline.
So while Madeline's getting her camera up, I'll just take a note very quickly about the organisms that I have up here on the screen, and you can see that our diatoms all have different types of adaptations that they use to be able to stay aloft basically in that upper portion of the ocean. So, their primary objective is to continue floating. So the activity that Madeline is going to do with the Great Plankton Sync Off is going to try to test some of these adaptations. So you can see some of the organisms that we have on the screen, and I'm going to go through Madeline's, I'll just click through them while Madeline explains her different models.
All right, so this is model A. We're calling it the star model. You can see it's shaped like a star. It looks a lot like a strinella. I'm not as great at pronouncing them as Katie is, so she can correct me if she wants. So this is our model A. Keep that in mind. I'll show it again before we sync them. All right, and then model B, we're calling this one the fuzzy spiral. This is actually Katie's model. This is the one Katie made, so keep that in mind too while we're syncing. But it looks a lot like Cattosaurus, which is the picture right there. It has a lot of like fuzzy projections in its spiral shape, so could lead to an interesting sync. And then our next one we're calling the pancake model, or model C. It's very flat. It's about the same width as the tube we're going to be syncing them in. And it looks a lot like Cosnodiscus, which you can see right behind it. And then our last model, we're calling this one the spiky circle. It's a little bit thicker than the pancake model, and it has these projections off of it. And we're actually saying it's a bit similar to Odentella. Yeah, that's our fourth and final model. All right. So I think, Katie, I'm going to go ahead and unshare your screen if you're okay with that. Perfect. All right, so the Plankton Sync Off, like we showed all the models and what plankton they're similar to. What we did is we took each of the plankton, had a little piece of Play-Doh we made them out of, and we had different craft tools that we used to adapt our different plankton, different wire, toothpicks, popsicle sticks, different things like that. So not like real plankton, ours can't move, and ours aren't made of the same materials as real plankton, right? So they are going to be a bit heavier. They're made of Play-Doh. So keep that in mind. With the adaptions that we're trying to show in these models will hopefully make the plankton sink slower. So why would the plankton want to stay towards the top of the water column? That's where the sunlight is. So they want to stay and float for as long as possible close to that sunlight so they can produce energy and get the food that they need. So we're going to keep that in mind while we make our guesses on which of these plankton models will take the longest to get to the bottom of our tube right here. So this tube, it's about 1 meter tall and it's filled with water. And we're going to drop each of the models. I'll have a timer on my phone, and I'll actually time how long it takes to get from the top to the bottom. We do have a poll to see if you guys can guess correctly which one you think will take the longest. So go ahead and vote. I'll show the models again. So remember, this is the star one. And then we have, this is the fuzzy spiral. I don't know if you guys can see that one okay. We also have the pancakes, the C model, and then we have our spiky circle right here. So just keep that in mind when you're voting. And I don't know I think everyone's voted, or a few people have voted. Keep on voting.
It looks like we still have a few more answers coming in. We'll give you just a few more seconds to add that. You are looking for the box with the green header on the top. And again, your options are A, star, B, fuzzy spiral, C, pancake, or D, spiky circle. And it does look like we have everybody voted, so thank you for doing that.
Perfect. Yeah, it looks like everyone's 6 votes, 60% of the votes is pancake, 30% is B, the fuzzy spiral. Okay, I'll show the fuzzy spiral again. And then we have 10% of the votes coming in as the spiky circle. So I guess we'll have to test it now, which is the exciting part. So I'm going to climb up here and I'm going to time how long each one takes, and we'll see who was right. First off, we have the star. Nobody voted for this one, so we'll see if it actually proves all of you wrong or not. All right. All right, that one took 4.37 seconds. So I think Ipsita is going to write down the times and put them in the chat box. I'm going to take the model out and then we'll run the next one. But I want you to keep in mind while I'm putting the models in and they're syncing, what about these adaptations that I've created or what pitfalls do they have that's going to make them sync slower or faster. And if you have any ideas about that, go ahead and put them in the chat box. And I think Kayla could read them out and we could have discussions about why they're syncing slowly or not.
Absolutely. And if anyone did this experiment at home before the program, we'd love to hear what kind of a model you made and how it might be similar or different to our educator-made models. And also, if you think of any changes that you might make, so for instance, for the star one just tested, ideas of how you might make it slower if she were to modify it and do a second test.
Yeah, I completely forgot to say that. Yeah, at the end, we'll do some modifications and see if we can make some even slower plankton. But next, I'm going to do the fuzzy spiral. We did have some people vote for this one, so we'll get to see how fast this one sinks or slowly it sinks. All right. All right, that one took 6.3 seconds, so a little bit longer. So it is a little bit more adapted to that slower sync that we're looking for. So far, everyone seems to be guessing correctly. I'm going to fish this one out. And Kayla, if you have any comments, please let me know.
Fantastic. I don't see any comments yet in the chat box, but just feel free to add them in there if you have any ideas. So that one was significantly slower than the first one. Anyone have ideas why? What might be different about this shape compared to the star? We'll let you ponder that and you can add it into the chat box as we go along. Oh, so very quickly we had a number of responses pop in that said more surface area.
Nice, I love it. Great response. Maybe that's why some of you thought the pancake is gonna go so slow. So this is our next one. We're gonna be letting sync the pancake design. And this was the crowd favorite. So we'll see if you guys are correct or not on how slowly this one will sync. All right. I don't know if you can tell, but it is starting to sink. It's just sinking very slowly.
We have a guess that it will take 8 seconds. So if anyone has any other guesses, we'll see who can get the closest.
I love that. It's been 14 seconds so far, so it is definitely sinking slowly.
It's already surpassed the 12-second guess as well. We do have a guess for 1 minute and 30 and a minute. 57 seconds.
Well, it's not even halfway there yet, and it's been 30 seconds. So.
49 seconds.
It's almost halfway there.
Okay, we have another updated guess, and based on our results so far, Nina thinks it will take a full 2 minutes and 15 seconds for it to reach the bottom.
Wow, that's a very specific number, Nina. All right, and it looks like it's about halfway down. The pancake is the winner so far, but you know, the spiky circle still could give it a run for its money, so don't count out that spiky circle yet.
We did so Luke was saying that he wondered if you put it in sideways in the future, if it would go faster. So I'm curious, Madeline, if you have thoughts on that, one way or the other.
I put it in instead of it being flat if I put it sideways?
Yes, exactly.
But right now it's completely like inside the circle of the thing. But I wonder if that would make an effect or not. It has been 1 minute and 34 seconds, in case you were wondering. And it is past the halfway mark now. So we are getting close to the bottom.
We have an updated guess for 4 minutes. I think that is our longest guess so far.
I mean, honestly, it could do that. Only time will tell.
I am curious what people think is different between the tube that we're using to attach and if these plankton models, if we were to go stick them in the Skidaway River or in the ocean, and how that might influence how fast or slow it's sinking.
Exactly. Well, there are a lot of different parameters out in the river that we don't have here. Like I said, we are using different materials, and the plankton is not able to move here. But out in the river, there's going to be a current, there's going to be, animals out there, there's going to be boats in the water making different waves. So it would definitely affect. There'd be other parameters, and there's different adaptations for plankton as well. We are close to the bottom now, and it's been 2 minutes and 41 seconds.
Thank you all for hanging in there with us. This plankton is far and away beating the other ones by leaps and bounds.
It is the front runner.
We do have a question about whether the level of salinity might make a difference.
I don't know. That's a great question. Katie, do you know? Or Kayla, do you know?
I would certainly assume that it does. They should be, it should allow them to be much more buoyant in salt water. So there would be an effect of the salinity as well. But that varies a great deal in the Skidaway River, as you know. So it just sort of depends on that. Other things that could affect it, of course, are some of the actual behaviors that some of these plankton show. So our models obviously can't move their flagella or do any of the other things that they would do, move water through them, etc.
And keep in mind, I used hose water for this. So they're, I don't know what I'm assuming it's fresh water coming from that hose. But our plankton has sunk all the way to the bottom. The final time is 3 minutes and 11 seconds. So that's pretty impressive, honestly.
And I do want to give a shout out to Catherine and their family, because they were the closest, coming in at 4 minutes with their guess.
Ooh, so we have that's a great guess. I mean, that was the closest one. That's awesome. All right, so I guess we'll move on to our final plankton. I'm actually going to have to fish this one out. Might take a minute. So if you have any comments about the plankton while I fish it out, please put them in, because this one's going to be hard to pull out.
So let us know if you have any questions. And Ipsita can go ahead and put into the chat box for you the directions for the activity sheet, so that if you want to do this at home, you might get ideas about what shapes you might try. I think it'd be really cool to try out using the same shape, but with different materials. So maybe you try a pancake made out of clay and one made out of aluminum foil, and see if maybe that weight difference makes any difference.
That's a great idea. And you could do it in a bathtub or a sink. I mean, you could float all of them down at once if you had that wider space instead of a tube. So that might be an exciting way to do it. We're going to sink our last one. This is the spiky circle. Looks like Odentella. Let's see how, I don't know if it'll get to quite 3 minutes and 11 seconds, but we'll still hope, and we'll let this one give its go. All right, that one was 4.77 seconds. So definitely not a front runner. I don't remember what all the times were. Do we have the timekeeper? Do they have the all the times written out? Who came in second, third, and fourth?
So Ipsita has kindly added them to our chat box. So Model A, drum roll, was 4.37 seconds, followed closely behind by Model D, 4.77 seconds. Model B was was much slower than A and D. It came in at 6 seconds, 6.3 seconds. And then Model C was 191 seconds, or a little over 3 minutes. So quite a difference in that race.
Yes, so we do have our pancake winner. It's a little bit messed up after having to fork it out, but it's still pancake. All right, so I guess we can move on to the portion of if anybody has suggestions about adaptations that I could make, anything I could quickly do. We could try making a fifth audience model based on adaptations to the 4 I have here. So let us know in the chat box.
So if you have specific suggestions for how Madeline can change Model A, B, C, or D, and even if we can't beat that really slow time from Model C, maybe we can make Model A, B, and D slower relatively. So we do have a suggestion. For Model C, could you add some holes to it?
The pancake, you want me to add some holes?
Yes, and see what happens there. I'll give you a couple of options, and then we'll let you choose kind of which one you want to do. There's also a suggestion that you could flatten out Model D, but keep the pipe cleaners. So make it closer in shape to the pancake, but keep those spiky projections. And then there was also, this one's cool. You could take that pancake and make it into more of a parachute shape. Or take that pancake and shape it like the hull of a boat. So those are the ones that have come through so far.
I think we probably have time to do 2 of them. So I'm flattening out the spiky circle, spiky ball we have right here into more of like a spiky pancake now. So I think that will be a good first one. We can see if we can make, what was the time for Model D originally? This one?
Model D was 4.77 seconds.
All right, let's see if we can get this one to sink a little bit slower than 4.77 seconds. All right. I've modified it. It is not quite as pancaked down as the actual pancake, but I've gotten it pretty flat. And I've kept the projections. So let's see how fast we can get this one to go. Or slow we can get this one to go. Oop, I might have made it too wide. Nope. Oh, there we go.
And we should add that when we're doing this at summer camp, any of the models that do not sync at all are disqualified. And the kind of the theory behind that is that yes, as a plankton, you want to sink slowly, but you do want to sink because if you're right at the surface, you're going to be exposed to air and a lot more extreme conditions than if you're a little bit below the surface of the water.
My pipe cleaners got too wide and prevented it. See if we can push them together. But yes, during summer camp, we actually have a paper circle each of the campers get. This is the exact diameter of the top of the tube, so they know not to make it any wider than this, which I should have used it when I was making my adaptations. All right, let's see if we can make this one sink. All right, that's our adapted Model D. All right, that actually did work. It was 11.6 seconds. So that adaptation actually did make it sink a little bit slower. Flattening it out, creating more surface area was the perfect modification.
That is awesome. That like more than doubled the slowness of it. Do you need any reminders of the suggestions that people had for other modifications?
I'm gonna fish this one out and then I can, I know we said we could add holes to this one. What was the other one that was suggested?
For that same pancake one, you could turn it into more of a parachute shape or make it more like the hull of a boat. So it sounds like either turning it down or turning it up.
Perfect.
And the other thing that's kind of cool to think about is that in addition to adaptations for how the animal or the in this case plant-like organism is moving in the water, can anyone think of a way those projections, those spikes, might help a phytoplankton survive in the ocean?
I'm changing the pancake right now.
Wonderful.
The parachute. I think that's similar to what Luke said earlier. He said he wanted it sideways, so we have a couple of people who were on the same thought right there. Try and show you what I'm doing on screen. I'm not that great with crafts, so this does take me a minute.
Well, and interestingly enough, if you really look at some of the real plankton, they are much more three-dimensional than just a pancake. So even the coccinodiscus is more sort of a pillbox shape. So it's got some depth to it. And others will kind of have some sides that are more cup-like. So they definitely have quite a bit of difference in their surface area on different sides. They're very three-dimensional.
Yes, they aren't made of Play-Doh either, so it does make a little bit of a difference. So I've made kind of a parachute here. I think this is generally what we're going for. I don't know if this will beat the 3 minutes, but we can see. So I'm interested to see how long this one takes to sink. So I'm gonna toss it in. It flipped, so it went in as a parachute, but now it is more of the hull of the boat. All right, that one took 7.45 seconds, so it didn't slow it down quite that much, but it is still pretty good time. It did take a while to sink. Are there any more suggestions for plankton?
That is a great question. I'm gonna scroll up and see if there was any that I missed, or if folks have additional suggestions, feel free to add them now. What happens, I don't know if you have any small pieces of pipe cleaner, but what happens if you add spikes to the top and bottom as well as the sides of your spiky circle?
Well, I would, but I kind of named the spiky circle pulling it. I used the end of my device as a fork to get it out of and I stabbed it a little bit too much. So I would try that, but I'd have to reassemble.
No worries. We do have another suggestion. We have a couple more suggestions that I can let you choose from. One is to make the star hole, the hole in the middle of the star one, smaller. Okay. So that one. The other option would be, what if you flattened out the spirals instead of spaghetti? So maybe flattening it.
Oh, so
flattening it like a coil, maybe.
I see. Yeah. Let me try making the circle a little bit smaller on this one, and if we have time, I can do this. This one has a wire in the middle of it, which makes it tricky to flatten.
Awesome. Well, thank you for modifying them for us.
Of course. I mean, I get to play with some crafts. I'm not great at it, but I get to play with crafts today, so what's not to love? All right, so here's the star, and I am making the circle a little bit smaller, creating more surface area. Maybe it'll float a little bit slower. What was the original time for this one?
Let's see. The original time was 4.37 seconds.
Okay. Maybe we can beat that. I'm going to pull the old one out, and then I'm going to put this one.
And I will say that usually when we do this activity at camp, we've got crowds cheering on their favorite model. So if you want to at home, you can cheer on the modified version if you'd like. And while you're fishing it out, we did have a question from the audience that says some plants, when they're left in the sun too long, will burn up and get too much sun. Is it possible for phytoplankton to get too much sun? And maybe Katie, if you can speak to that piece.
That is a really good question. I think when you think about plants sort of burning up, or that sort of transpiration, losing all of their water from their cells is why they sort of burn up. And our phytoplankton are aquatic organisms, so they're not really going to have the same kind of effect if they're they're not left high and dry. So certainly if they come in with the tide and they are left with the outgoing tide, then they're certainly going to burn up. But I don't think that they're going to burn up just because they're on the surface. Although if there's a bloom, again, you're creating almost a high and dry situation. So I assume in a bloom that could happen. Most of the time, there aren't enough of them to be that exposed, however.
That was a great question. I am going to sync this now. What our original time was 4.37 seconds, 47 seconds. I'm forgetting already, but maybe we can get it to 5 seconds.
4.37 seconds. That is the time we are trying to beat. And by beat, I mean we are trying to get slower than 4.37 seconds. So drum roll, please. At home, not Madeline, her hands are busy.
It must be 6.2 seconds. Wow, everyone's doing great with suggesting these adaptations. You're a lot better at this than I was with making them. But I guess increasing the surface area, eliminating that hole made it, sink a little bit slower and gave it a better time.
Those are some great suggestions from the audience. And we had another hypothesis from John that if the pancake was in a wider column of water, it might sink faster. So I'm wondering, Madeline or Katie, if you could talk a little bit about how, and someone else had asked this too, like if pancake is so much slower, why aren't all of the plankton shaped like a pancake? So I'm wondering if you could talk a little bit about what some of those other features, the spikes or the chains, how that might help the plankton thrive.
I think Katie can add to this. I think the pancake does a lot better here than I think it would in the real world because it is completely filling. It is maimed now, but it is complete. This is the width of the cylinder, and it's when it was flattened, it was completely the same width. So it is. That's why it's so much better adapted for the tube rather than the real world. But it does give students and campers a great illustration and example model of like what adaptations work, which ones work well, which ones don't.
Yeah, I completely agree with you, Madeline. And I think some of the things that we talked about, plankton aren't made of, Play-Doh and, heavier, kind of denser things. They're made of silica and cellulose. So those are lighter materials. They are porous. They have pores for water to come and go, so they can actually change osmotically, how much water they have in and out of their cells. They can move water through their cell. They can use, their flagella to move themselves if there are upwellings and changes as far as the temperature of the water, the salinity, the currents that are out there. So there are so many different factors that are going on to, out in the real world, as opposed to our very constricted column of water. So, we are basically blocking all of that laminar flow. When we're putting something in there that almost is the same diameter as the tube, then there's no room for that water to move past it. And that's just not true when you're out in the, in our rivers or in the ocean, certainly. So I don't know if that spoke to it or not. Some of the projections can also, they can capture air bubbles, and, some of those sort of hair-like structures can actually, they'll hold on to each other, and they're kind of raft on each other. So that surface area can actually change for those organisms as well. So some of those little projections can allow them to kind of cling to one another, and you'll get large, groups of them together, or creating those little star-like structures and that kind of thing.
Well, thank you both so much for sharing your knowledge about plankton and letting us experiment and test out some different models with you. It is just about time for us to sign off. So before we go, I would encourage everyone to fill out our 1-minute Qualtrics evaluation. It only takes a minute. I know that some people have been to multiple of our programs, but because the content is different each week, we still value your feedback every week. So that is in the chat box. And I would add that our chat box will disappear once we sign off. So if you want to go ahead and click on that link now, or copy and paste it, and you may want to do the same thing for the activity directions. All of those you can download from our website, from the event posting for today's program as well. They're posted up there. So we'll give folks just another minute to copy and paste if you need to. And we do have a couple of minutes if there are any last questions that people have about plankton. So we do have a question about how old do plankton get?
Oh, that's a really good question. They're constantly renewing themselves. So their life cycle is fairly short. They're only going to live for, a few hours to a few days for an individual organism. And then they'll reproduce pretty quickly.
Thank you, Katie. We also had a question specific to the Qualtrics evaluation. Someone asked, is it the same link for all of the programs? It is. And the way that you can let us know that you're evaluating today's program as opposed to last week's is that very first question will ask you for today's date. So any format you want, you can write it out, can numbers in, but just let us know that it was June 23rd that you are evaluating, and we will get that feedback and we will incorporate it and value it greatly. So I think those are all of the questions that we have. If people have additional questions, all of our contact information is up on our website, and I would encourage you to reach out with any additional questions you have. And if you had fun, I hope that you will join us next week. Next Tuesday, we're going to be doing a fish dissection, so we'll be in another one of our labs, this time our fish lab. And then on Thursdays, if there's anyone younger in the audience, we do have programs for 4 to 8-year-olds. This Thursday will be all about fish. We'll get a chance to meet some of the fish in the aquarium and ask the important question, are you a fish? So join us on Thursday if you're interested, and otherwise have a wonderful week. And we'll see you later, alligators.